Emily, and thank you to Healthcare Workers for Climate Action for all the good work you do, and to Tick-Free New Hampshire and their work they do on outreach across the state, as well as BeBop's lab. As Emily mentioned, I'm on the advisory board for Healthcare Workers for Climate Action. I've worked in public health for 3 decades in New Hampshire and the Northeast US, mostly in the area of environmental health, indoor air quality, and climate change. As we know, the Northeast is getting warmer and wetter on average, with more severe weather and sea level rise, and these changes all have implications for population health and clinical care, which we'll hear more about tonight. The problem of ticks is near and dear to my heart. I'm sure many of you, anybody who has pets or kids, when I moved to New Hampshire in the 1980s, there were a few ticks in central New Hampshire, although a growing population on the seacoast area. Today, we have an abundance of ticks across southern and central New Hampshire and expanding into the northern regions, and we'll hear more about that today. We're going to move on to our presenter and the topic at hand. I think this is likely the shortest bio I've ever read, and that's a compliment to our speaker. I've worked with Kaitlyn on Tick-Free New Hampshire for a number of years now, trying to promote the issue of improved tick surveillance and especially outreach and prevention. Dr. Kaitlyn Morse is the executive director and founder of BeBop Labs, a 501c3 that focuses on gathering information about how climate impacts personal health and disseminates the information. Dr. Morse is also the CEO and founder of VaxSyna, a biotechnology company making vaccines produced in plants. So, Dr. Morse, please take it away. Tell us any more about you you'd like to say, or on to our presentation.
I'm just going to start sharing my screen because I have a slide about myself. But for some reason, I lost how to share my screen when I did that. Do I still have the ability to do that, Emily?
You do, yeah. Do you see the green share screen?
Oh, here it is, yeah. I needed you to tell me that it was green, thank you. Okay, I might just leave it in this format if that's all right. If you guys can, if all of you can see that. There we go. Hopefully you can see the whole screen. I moved it. I can make it smaller or bigger if that's all right. Okay, so we already went over the title. So again, I'm Dr. Kaitlyn Morse. I grew up in Vermont. I've lived in New York, Washington, Connecticut, Mass, and now New Hampshire. I'm a mom. I love the outdoors. I love hiking, single track mountain biking, swimming, running, gardening. I was a studio apprentice at a ceramic studio to try to figure out if science was the best way to change the world or was it art. And I found that maybe both. I'm one of those people who like to make the world a better place. I have a PhD in immunology and infectious disease focus. I created a vaccine against anaplasmosis during my PhD training, which was in 2006 to 2010. Thus, I've been learning about ticks and tick-borne diseases for over 17 years. I am an entrepreneur, I'm a serial entrepreneur. I've co-invented a laser adjuvant when I was a postdoc at Harvard University down in Boston. I'm a founder and executive director of BeBop Labs. I've created a spin-off company called Morse Innovations, and I am now a founder and chief executive officer of a biotech company called VaxSyna, making vaccines, which is definitely been part of my 10-year goal after getting my PhD. So it's another big passion, but BeBop Labs is definitely a passion project. It's a 501c3. Its mission is to track impacts to health. Just so happens that I started focusing on ticks and tick-borne diseases because I realized there's a gap in New Hampshire and wanted to fill that. And we've been staying with it because it's a volunteer-run organization. All the boards volunteer, including myself. So it's a matter of time and priority. So ticks and tick-borne diseases are a priority. And where we just want to continue to grow and expand. It's a big problem, so we put our focus there. Like I said, BeBop Labs could track any impacts to health. It's formed in a way that there's more space to grow. So it's a 501c3, so it's a community organization. It doesn't belong to me as a founder. It doesn't belong to anyone other than a community. So our vision is to foster scientifically muted community, which involves tackling these impacts to health with hands-on projects and innovation. So that's part of how we govern the mission. You can find more about BeBop Labs on our website, on the Facebook page. We're very active with social media. One of my board directors monitors that. I don't personally. You can email me more at info@BeBopLabs or my Kaitlyn@BeBopLabs.org. And we also have a mailing address. So the overview of this presentation is really focused on ticks and tick-borne diseases. I go through a schematic. This is the schematic that I've come up with to talk about the tick landscape, starting with awareness and then prevention of ticks, surveillance, diagnosis, and treatment. Surveillance is key because it's in the middle. All the information from surveillance of ticks and tick-borne diseases actually feeds into these others with awareness, prevention, diagnosis, and treatment. You need to have the surveillance in order to do all the others. That's why it's in the middle. Everything hinges on it. Notoriously, BeBop Labs is one of the only organizations in New Hampshire that tracks ticks and tick-borne diseases. Although, as of 2023, there are several other projects that are happening in the state that include surveillance. We just have been the only organization for a while, since 2017. So what we do is we crowdsource for ticks. It's called passive surveillance. It's not actively flagging for ticks, it's passively gathering ticks. So if someone comes across a tick, then they gather information about that tick, put it in the mail, and send it to BeBop Labs. I get, during heavy tick season, I can get hundreds of letters in the mail. And we have an open mind of like what ticks come in. So this is very much passive. It doesn't matter the species of the tick, where it was found. We just need that information. So we ask for date the tick was found, the number of ticks, the location, as specific as possible. So sometimes we get longitude and latitude coordinates. Sometimes we just get an address. So we try to get like as close to longitude and latitude as possible. That can be a caveat with passive surveillance, was location is one of those things that can be challenging, which is why putting passive surveillance together with active surveillance will help mitigate or help understand how climate change impacts ticks a little bit more. So location's a little bit of a caveat with the way that we gather ticks. The activity the individual is doing when they found it, who it was found. We gather ticks from humans, goats, fence posts, tables, rugs, it doesn't matter the host. We just want to know if it was biting or crawling even. And then we ask for more information if it was biting, because after a tick is mailed to us, then we identify the species of the tick, the stage this tick was in, if it was engorged or not. And then we also test them for disease. So we test primarily black-legged ticks, Ixodes scapularis, for the most prevalent tick-borne pathogens in New Hampshire, which is Lyme, anaplasmosis, and babesiosis, which I actually have the scientific name for right here. And the dog ticks are Dermacentor variabilis. On top of that, we test for Rickettsia rickettsii, which is Rocky Mountain spotted fever, as well as Francisella tularemia, which is tularemic fever. So I'll go a little bit more into detail of the diseases later on that we test. This is just a kind of an overview of the way the project and way BeBop Labs is run. And because of all of this, we've actually gathered enough data from 2018 to 2021. This paper was, we gathered enough data from the Community Science Project where we published a peer-reviewed publication of all of our findings and put that in. It was released of May of 2023. So my overview of this presentation is actually a primary focus is on this article. BeBop Labs paid arm and a leg just to make it open source. So this is freely accessible. You can download it, open it as many times as you want, read it. So a lot of the findings that I'll talk about today are focused on this, but again, we're going to go over that landscape, awareness all the way to treatment, and surveillance being, key, what this paper is written. Okay, so starting right with awareness, I always talk about the key pointer that any presentation that I ever give is how to identify a tick. BeBop Labs primarily gets black-legged ticks and dog ticks in the mail. So a black-legged tick, other known as deer tick or Ixodes scapularis, and dog ticks, other known as wood ticks or Dermacentor variabilis. You can tell the difference in these species by all sorts of things: size, color, hosts, time of year that you find them, where in the wilderness you find them, their questing patterns, what diseases they are vectors for. All of those are different ways to identify a tick species. That's why it's the most interesting and the most important thing that you need to know about ticks in New Hampshire. Just looking at these pictures in general, the black-legged tick, the female is red, smaller than the dog tick. The male is black, smaller than the female dog tick. The dorsal shield on both of them is black. These are adult ticks. The nymphs are much smaller. The next slide will be on the size of the nymph, deer tick. The dog ticks are brown with white dorsal shield. The male has a white dorsal shield but a mottled back. They're bigger in size. They're more brown, and you can see they have 8 legs. They are in the arachnid family. You can also look at their mouth parts. So the mouth part on the female deer tick is really long, whereas the dog tick is relatively short. If you're looking at other species of ticks, like not in the Dermacentor or Ixodes genus, and maybe in the Homophilus genus, their mouth part might be different. So the Homophilus is Asian longhorn tick. And the hosts, so in the research that BeBop Labs has done, black-legged ticks are very much actively seeking human hosts, whereas dog ticks have a weird questing pattern. And you'll see that later. They quest everywhere and are more opportunistic in their questing. Black-legged ticks seek out that blood meal. The time of year, you'll see later too that they have overlapping patterns, but there's a time in the fall where black-legged ticks only come out. And in the wilderness where they are, they're a little bit of correlation with dog ticks liking more water, but I haven't figured out a statistical method to like really show or prove that hypothesis. It's just one of those observations I have, and I'm still trying to figure out how to scientifically test that hypothesis. I need the right expertise to really figure out that right now. But that's kind of like one observation, and so I kind of mentioned it, but you'll see later on that it may not have a lot of validity. We'll look at some maps. So here's that picture of the nymph tick that I was talking about. The black-legged ticks, the nymphs only have 6 legs. When they get a blood meal, they'll shed their exoskeleton, and what I call leveling up to an adult tick, they'll then get 8 legs, and they're tiny. They're the size of a tip of a pen, poppy seed, sesame seed. All of those are different sizes of a black-legged tick. You can't tell the sex of a nymph tick. It has to be tested. The males and the females look exactly the same at that stage. They're even almost see-through. So it's like a crawling freckle. So it's very challenging to spot those guys. And the hardest part, I always say, is actually finding that tick. So moving on to prevention, I always say a tick tech a day keeps Lyme away. It's the best way to keep yourself protected from the tick. The hardest part, again, is finding the tick. So if you go and look for ticks on your body or on your pet, they're going to like warm, hairy, and moist areas of your body. Ticks are attracted to butyric acid and carbon dioxide. So when you breathe out, they can smell that. Butyric acid is the chemical that's released in your BO. So you can actually trap ticks by using dry ice, and dry ice releases carbon dioxide like you breathe out. And so you can have a carbon dioxide trap or a butyric acid trap. You can make your own butyric acid. I've done that before with potatoes and butter, and you can like produce that nasty BO smell that ticks will then go in and get attracted. So you can trap ticks that way. So you can find them in areas like when I've actually changed my heat patterns have changed since I become a mom and was pregnant. before becoming a mom, I wasn't a tick magnet, it was my husband. And now that my temperatures have changed and my body hormones have changed, I'm actually more attracted to ticks than my husband. So it's part of like your growth and development cycle as a human that your temperature might change. And so you look for younger ticks, like anywhere below your knees and down to your ankles are the nymphs. They're crawling up, questing. The adult ticks will be above your knees, up into the bottom of your hairline. They like to hide in your hairline. They'll move and they'll crawl more up to hide in different various places. So your back, chest, armpits, groin, you'll find them there. Likewise with dogs, I mean, behind their ears, armpits, and their crotch. So they'll hide along their legs too. I like this picture so we've made this to 8 different methods to prevention tips. It's standard wear. So the clothes that you wear, lighter colors, so you can find ticks, shoes, socks tucked into your pants and your shoes so that you can find them. They don't ever get attracted to the skin, like you hiding your skin down there. I spray my shoes and socks with permethrin, a 0.5% permethrin. That is a approved pesticide that does not stick to the epithelial layer of your skin. It only sticks to fabric. So it's better if you use permethrin on fabric only. If you're going to spray your skin, you're going to want like DEET or lemon eucalyptus are approved pesticides to repel ticks. Lemon eucalyptus just has to be reapplied every 2 hours. Also, this has information on here about like stay away, walk in the middle of a trail, stay away from vegetation where ticks quest. Ticks lay their eggs in leaf litter. So if you're on the border of a forest into a field, there's going to be a bunch of leaf litter. That's likely where you're going to find a tick. So you kind of want to stay away from that area during a peak tick season. And then just perform your daily tick checks. There's also prevention methods about landscaping around your house, creating a tick safe zone, putting out wood chips that repel an oil. So ticks breathe out of their legs. So if you can clog their breathing pores with an oil, you can suffocate them and die. They'll die. So things like a wood chip that gives off an oil, like a pine chip or a cedar wood chip, that will give off that oil will put the oil into the pores of the tick's legs and they will suffocate and die. So that's a good way to like keep your pollinators happy and only kill those crawling insects with pine. Likewise, like tick tubes are the same kind of concept where you spray a cotton ball. I actually have that. I might go back one slide. So you spray cotton balls with permethrin. I use a higher percentage of permethrin when I make my own because the idea is you spray the cotton balls and a rodent will take that cotton ball and bring it to their home, their nesting area. And then they will rub their bodies in the, pesticide treated cotton ball and kill all the ticks. So idea is you hide these cotton balls shoved into toilet paper rolls in places where you find a rodent, but you want to be careful with aquatic life because permethrin kills the aquatic life. You also want to be careful with cats. If you have cats that are indoor outdoor, permethrin can cause a neurological issues with cats. So if you do have a cat that's indoor outdoor, you may not want to use permethrin. You might want to use befedrin or another one that is, I'm losing my mind right now, I'm forgetting it, that is good for cats. So going back to the landscaping zone, the other thing that you can do is to leave ferns in that are out. Ferns are an ancient plant that have evolved to have a natural pesticide. If you notice when you walk through a woodsy area with ferns, mosquitoes are down, and ticks will also be down. So ferns act as have a natural insect repellent. So we did this one. So what do you do if you find a tick? If you find a tick biting you, you don't want to put any grease, peanut butter, you don't want to burn it. Ticks are vectors for viruses, and as you know, viruses can go airborne, so burning something doesn't sound like a good idea to me, someone studying viruses and diseases. So if it's embedded in you, you want to use something that allows you to grab it as low as you can possibly and pull it up straight. Don't want to twist it, move it. BeBop Labs did make these removal tweezers, like stainless steel, that can be sterilized, and one tip is designed for smaller ticks. The other side is designed as a scoop for larger ticks, just so that you can pull evenly steady pressure upward straight direction. Afterwards, you can clean the area and like use rubbing alcohol or soap or water to clean the bite area to help it heal. But I also advocate for saving your ticks. So identification is the most important. As you know, identifying the differences between the black-legged tick as well as a dog tick is also helping you identify which possibility of your risk of getting a transmitted disease. So saving that tick, and since BeBop Labs is a community science run organization, we're constantly researching ticks, is in the state, you can send it to us, and we get more dog ticks than we do black-legged ticks, just because that's what happens in New Hampshire. But if you're worried about getting a transmitted disease, I would not send it to BeBop Labs. I would send it right away to a tick testing lab. We partner with MedZoo or Tick Report to get ticks tested right away. It does help with the peace of mind for disease diagnosis, but it's not a diagnosis because you're testing a tick, not your system. So it's not a diagnostic test for you contracting a disease. And I'll get more into that later, but this is just how to remove a tick and what to do when you get a tick. So now getting into the meat of the presentation, talking about surveillance. So like I said, BeBop Labs took 3-4 years of data. We're continually collecting it. 2018, 19, 20, 21, 4 years of data to gather the ticks and where and when, and we published it. So we were agnostic about the type of tick species, the host, which means this publication is very much centered around One Health. One Health is a collaborative effort that involves getting health and obtaining health information for people, animals, and our environment. And so that's why it's defined as a One Health. It's very important in epidemiologically studies to do a One Health methodology to control diseases. So in this study, we gathered ticks from 2018 and 2021. We received unprecedented numbers of ticks, which makes this a very robust study. The number of black-legged ticks that were collected in that period of time were 4,257. The numbers of dog ticks that were collected were 9,876. In the male, that's only for the state of New Hampshire. Other studies that are out there don't have this One Health approach. They look at humans, they look at only black-legged ticks, they're looking at the entire United States. They collect less numbers than we have. The host data, there are various different hosts that we got it from. Black-legged ticks, there wasn't really well documented. It was mostly unknown hosts for black-legged ticks, and mostly the dog ticks were found on humans. Dog ticks were also found on more inanimate objects than black-legged ticks. And we gathered ticks from all over the state, but Coas County was very underrepresented, which you can see in our maps. So on the left is the black-legged ticks, or Ixodes scapularis. On the right is the dog ticks, or Dermacentor variabilis. We kind of we put the scale at the same numbers roughly, so that, yeah, exactly the same numbers, so that you can compare the two species. You can see that we got more dog ticks than we got black-legged ticks, and dog ticks are kind of all over the state. and not really following a pattern. If you lowered the scale on the black-legged ticks, you'd find that there's kind of a pattern where the people are, like following 93, the centers, the town centers, Manchester, Concord, left here where like Hanover area, and then Rockingham, Hillsborough County, where there's more people for black-legged ticks. This is a map that's not from the publication. It's just the BeBop Labs only map, but you can look at the publication map too, and you'll see very similar. I put this one up because it shows that we did get some from Coas County, but very little. There's very little, we don't know reasons why Coas County. I'm actively searching to get more ticks from that county to figure out, what's happening up there. But like I said, an increasing amount of evidence correlates to climate change, to increased number of ticks and changing seasons. So this is one of the figures from our publication that describes the time of year that ticks come out. And I wrote in observations about these graphs. So I'm going to go through how to read them. On the left axis is number of ticks. On the y-axis is the week of the year, and then also bracketed into different quarters of the year. On the top is black-legged ticks. On the bottom is dog ticks. So what you can see is that black-legged ticks come out earlier every year. So every line is indicated of each year. So 2018 is blue, 2019 is green, 2020 is gray, and 2021 is yellow. And so for black-legged ticks, the exponential incline of when you start to see the tick is actually earlier every year. They're higher in numbers in the spring peak, peaking around May, and then the fall peak, peaking in October. There's less there, and it's more variable, so it's hard to see a pattern. Year to year, the numbers of black-legged ticks about stay the same. And there's an overlapping peak in the June time of year for dog ticks and black-legged ticks. Dog ticks look like they come out every other year in more abundance. And that was also confirmed in a table of the publication as well, where you can actually see the numbers from year to year. But looking at this, you can see like the blue and the gray lines are less than the green and the yellow, showing that they only come up every other year. We also have a picture or a figure from the publication that shows human activity. I didn't present it because it wasn't really well high numbers. We didn't get a lot of human activity association, but it did confirm this bimodal distribution of black-legged ticks, as well as that it's a higher risk to find a tick when you're hiking, walking, or doing gardening, yard work, or backyard activities, and lowest risk for biking, and for when you're doing biking to find a tick. And dog ticks are found inside more often than black-legged ticks, which agrees with the host data, showing that it has a weird questing. Dog ticks have weird questing patterns. This one I'm gonna have to show my slideshow for, because there's the transition. I have an animation for the thing that goes over it. So we also presented the percentages of ticks that were found to have diseases, and this is only for Ixodes scapularis. Out of the thousands of dog ticks that we found and tested, only one of them was positive for any disease, and that was actually Francisella tularemia. And as from the questions that I ask when I get a tick sent in the mail, there, can you see that better? So we only got one Francisella tularemia positive of dog ticks. So that's a very low percentage of dog ticks that carry disease in New Hampshire. For the black-legged ticks, we find about 40% of them carry Borrelia genius, which correlates to 36 to 40% of them carrying Lyme disease. So about 37%. 1% are Borrelia miyamotoi, which is a Lyme-like disease. 6% carry Anaplasma phagocytophilum, anaplasmosis. 5% carry a Babesia microti, which is babesiosis, a protozoa. And 7% of the ticks the black-legged ticks that we tested were co-infected with more than one pathogen. So Lyme plus another usually. Oops. There we go. So this is an easier way to see that. I just showed you from year to year on that table. And to summarize that, overall, 37% ticks carry Lyme, 5% Babesia, 6% anaplasmosis, and 1% miyamotoi. Okay. All right. I like presenting this way. It's a little easier. So I'm going to keep going. So I did look into the surveillance data and how it's important to model how climate impacts health. And in terms of ticks, you can track how the surveillance models the tick range expansion, as well as disease exposure. Like very high temperatures drive ticks to stay in nests longer, resulting in reduced tick-borne pathogens, and that happens in the southern US. Also, it changes climate impacts tick life cycle and transmission cycle changes, such as like the temperature affects the number of eggs that are laid by engorged females, the egg development, the development of larvae and nymphs, the questing nymphs in adults, as well as the warmer the climate, the shorter the life cycle, thus more larvae survive to be reproducing adults and thus seeking more blood meals. Tick questing or human activity changes. So the length of the time a tick quests during the day can be affected by climate and temperature, and also the time of year, increasing the host and tick interactions for different activities are ways that climate impacts ticks and thus your health. So there is a number of, these is slides showing different publications and trends. I did a quick publication search for this talk to show the climate trends in literature. So each of these bullets are a different publication in the title and where you can find them. The first one is showing this image here, is an overview of complexities to the tick life cycle and transmission cycles that may be affected by climate change. They did project that tick distribution will expand poleward, upward, and upslope altitudinal expansion, as this figure indicates, but less abundant around the equator where very high temperatures, and that's connected to like temperature, latitude, altitude, rainfall connection, and habitat suitability for tick species. The next paper, Elmaya, is a review on tick expansion into Canada, considering temperature, land use patterns, parks, deforestation, and federal policies. And this Cooper et al. Paper shows an 18 years of annual county level of Lyme disease data using to investigate prior effects to climate variation on disease incidence. And the strongest climate disease association detected in this paper was between warming annual temperatures and increasing incidence in the Northeast. That's New Hampshire. That one's a very well done study. So I also wanted to show up common myths and facts about how ticks, basically about ticks. So one myth is that dog ticks carry Lyme disease, but in observations to debunk that fact is that one, BeBop Labs never found a dog tick that carried Lyme disease, and two, there's a number of publications out there showing that Dermacentor variabilis dog ticks are not competent vectors for Lyme disease, Borrelia burgdorferi. And the next myth is that ticks come out during the warm season and they die off during the winter. BeBop Labs has collected black-legged ticks from all months of the year. I showed that slide of the time of year from the publication. Every single month, we've gotten a letter in the mail and containing a black-legged tick. And the tick physiology is designed to survive lower temperatures, such as they contain an antifreeze glycoprotein in their hemolymph to survive sub-zero temperatures. They lay eggs in their leaf litter and use the leaf litter and animal nest during the winter to keep warm. The mortality of unfed ticks in the winter is actually the same as during the summer. And also, ticks can rehydrate by hydroscopic saliva by secreting the hydroscopic saliva, that's saliva from humidity, into their mouth parts, their hyposome. Okay, so moving out of surveillance into diagnosis. The next two parts are very quick. For diagnosis, it's actually to test your body for tick-borne disease. Usually it's extracted and taken blood and done an antibody test via ELISA or Western blot. That's only 60 to 80% accurate depending on the test and the stage of the disease. It's less accurate if it's an early onset disease. The test can take up a week to get results. There is, post-COVID, there is more of an ability to do PCR testing, which is more of a direct diagnostic test of the bacteria in your blood rather than the immune response to the infection. And that is way more accurate. So a PCR test of your blood to identify tick-borne disease is probably the way to go. I'm just unsure if New Hampshire has the infrastructure or the policies in place to do that test. We tend to be a little bit behind that way. The accuracy of testing a tick is 99.9%, and it can be done in 3 days. It's not diagnostic, but it's a peace of mind knowing if you got bit by a black-legged tick and it didn't carry any detectable diseases. That's nice to know. And here's a slide describing the length of time that it takes for a tick-borne pathogen to transmit from ticks to humans. I get these questions all the time. There's a number of different publications that I tried to outline underneath each of one of these bullets. I probably won't go on through the publications. I'm just going to go through generally what the results are, but I'm not saying that any of these publications are perfect. And there's definitely more research that needs to be done, because in our lives, we don't know if we get a tick that is biting us, if it had a previous blood meal before it bit one of ours or our family members. So it could have a prior, like it could be bit a mouse and then latch off and then bite me, and thus it would have a transmission time shorter. So for Anaplasma phagocytophilum, it's generally considered about 36 hours or longer for 70% transmission. For Lyme disease, it's greater than 24 hours. I usually say 24 to 48. There's a difference between if you have one tick or multiple ticks, though. And these weren't perfect studies where like the minimum amount of time they looked at were 16 hours, or they only look at nymph ticks rather than adult ticks. So generally, because Borrelia burgdorferi is a spirochete, it takes a little bit longer for transmission. But I would say, yeah, again, all of these are still need to have further follow-ups to like control for all the variables that we see every day. For Babesia microti, 24 hours, but that was the minimum time looked at. So it could be less than that. And for Powassan, it was very successful at transmission of 15 minutes, but 15 minutes was the minimal time that was looked at. It could be 15 seconds. It's a virus, so they tend to transmit a lot faster. The diseases, so tick-borne diseases are required to report in hospital setting to the Department of Health, but they're severely underreported and under-tracked. So if you look at this table, I culminated all the data from the Department of Health that was tracked for Lyme disease, anaplasmosis, babesiosis, and Powassan. So these are human cases that were reported to the Department of Health. And some of them have asterisks showing they were estimated numbers. And some of them haven't, like one of them has a star saying incomplete data for that one. But also if you look at the trends, when you graph it, it doesn't have a really good pattern. And you would probably estimate from like the tick data that I've been showing, that there's more of a trend. And so it's, yeah, I think it's still a work in progress, even though there's 10 years of data. There's, like anaplasmosis has the best trend that you can kind of see, but I would say the CDC predicts that tick-borne diseases are 10 to 12 underreported, but I'd say for New Hampshire, they might be 30 times underreported, just from being in this field and having a network of 80,000 people. You kind of get word of mouth who has what. And so just from like my knowledge of being in this field, I would say it's way more underreported. So moving into treatment, I wanted to give one slide on this because my expertise is in vaccine and treatments. So there's a difference between treating Lyme disease, in particular for early onset versus chronic. Doxycycline is a very effective antibiotic for early onset Lyme, Babesia, and anaplasmosis. But there's also a thing called post-treatment Lyme disease, because the spirochete can hide from your immune response and from treatment in places that it's hard to reach for antibiotics, like your muscles, your bones, and your brain. And thus the symptoms are like arthritis, muscle fatigue, and brain fog. So they create a biofilm that's like a protective castle for bacteria, and it's really hard to penetrate. So in addition to like doxycycline, there's other therapeutics that are being used. Common ones, actually, the herbal remedies, there's many of them that are used successfully to attack the biofilm first and then used in conjunction with an antibiotic. And sometimes that's doxycycline, but sometimes alternative antibiotics such as azlocin or disulfiram, which are very successfully used in humans to treat Lyme disease and other tick-borne diseases. There's plenty of antibodies in preclinical models right now, or that are developed like monoclonals, but they're not of them are approved for humans. Likewise, for vaccines, there's only one tick-borne disease vaccine that's approved, called Ticovac. That was approved in 2021 against tick-borne encephalitis in Europe. So for traveling abroad, you can get vaccinated against tick-borne encephalitis and protect you when you travel abroad. There are a couple of different vaccines in various different phases of development against Lyme. Valnevia, or VLA15, is against Lyme and in phase 3. Pfizer helps run that, but it's been a very mismanaged clinical trial. Lyme prep antibody, which was developed actually from University of Massachusetts, Amherst, I believe, came out of that, lab out of there, and that's in phase 2. And then there's many variations of a vaccine using tick spit that are in preclinical trials. There's a very promising one out of Yale using mRNA. And the idea is it's a vaccine against the proteins in ticks that they spit out, so it could vaccinate against all of the tick-borne pathogens that I mentioned today. There's actually 28 of them, different pathogens that a tick can carry. And if you vaccinate against the spit, you won't get any of them because your body will respond to the spit, and kill off anything that goes with it. And so thus it works for babesiosis and anaplasmosis as well. So I'd like to thank my board of directors, and that's all of these people listed here. We've had several interns that helped gather the data. I collaborated with a group out of Spain to develop that publication. They did a lot of the climate change modeling that was published. We have various different partnerships with different organizations like New Hampshire Camps that have gathered ticks and done the community science. So the community scientists, I need a very special thank you for anyone who's ever collected a tick and mailed it to BeBop Labs, because they make it possible. And through them, I've had so many different stories. I actually built a spin-off company out of BeBop Labs because when I involve the community in hands-on science, which is like collecting ticks, they innovate and they try to solve the problem. So I actually made another company that I have already dissolved because of the pandemic. that I can get into if you have questions about it. But I made another company that was allowing individuals to innovate and patent and make solutions to the tick problem along treatment, diagnosis, surveillance, all sorts of things, how to kill ticks. It was really cool, and how people were very interactive innovatively when they just collected ticks. And I also work with a group out of Dartmouth, which is doing more surveillance and going to use more of BeBop Labs passive surveillance data to track climate change and how it impacts ticks and hosts and all sorts of things. And so thank you. I hope you have lots of questions. You can always mail me ticks. I'll learn about you that way. It's interesting how I learn about people via the mail. We are a 501c3, so you can donate to support BeBop Labs on our, usually the money goes to providing free tick testing. We try to get the data back to people as we can, but we don't have a guarantee on how long that takes. So I'll start sharing my presentation now and take any questions.
Thank you, Dr. Morse. Kaitlyn, that was great. We appreciate the wide-ranging topics you covered. And as we're going through the first, and I see maybe a second question there, we'll encourage people to look at the chat function and go in there, put in a question, put in a comment if you have it for Dr. Morse now. So the first question that came up was, is there any data on what percentage of people with Lyme disease ever found a tick on themselves?
So that's a two-sided question. There is the percentages of ticks that carry Lyme disease, and there's the percentage of people that have been diagnosed with Lyme disease. It's hard. I guess actually, because BeBop Labs has the data where we can analyze the data very particularly where we have a tick biting and whether they're biting or versus crawling, and if they're positive or negative for Lyme disease in general. But that analysis wouldn't be as successful as just analyzing a tick that carries Lyme and analyzing a human that was positive, because there's a hard link between transmission. So what we found with ticks that are positive for Lyme disease, about 40% of black-legged ticks, 2 out of 5 carry Lyme. And it's hard to say with the diagnostics of humans, how many are actually positive or negative for Lyme and who've been bitten versus not, because the diagnostic test isn't very accurate. So I don't have a good percentage on the people side of it, and it's underreported too. So that one's a harder one to answer for the human side.
All right. Well, I've got a follow-up question because I'm always curious. When you get your reports back, where is the most prominent place they found on it? You mentioned earlier that the earlier stages of the ticks are usually found in the lower part of the body, and the adults are found higher up. But where is the most prominent bite point? Because when we're applying not pesticides, but we're applying repellents or putting permethrin on our clothes, I'm always curious, like, what's the most effective place to treat? So tell us about that.
I don't think there's information out there. So BeBop Labs has a little bit of data on that, but it doesn't have a strong enough end value. I know Tick Report gathers that information as well. But if you culminate all of that information together, there's not a lot of patterns there. It's just, there's not a lot of, there doesn't have a lot of flow to that, or it hasn't been analyzed correctly to answer that question. But in my view, when you culminate all of that information, there's not enough N value, so a statistical value, as well as there's no pattern to really answer that. Sure. So I think the answer is we don't know.
There's a study done with permethrin treatment of socks and shoes and people wearing shorts, and they found that if you did permethrin treatment of the shoes and the socks, that had significant reduction in the amount of in a tick who was put on the shoe and crawled up, and even shorts and t-shirts had some protective value. So I was surprised that even without a full set of clothes, which is very hard to put on hot days, especially if you're doing a lot of work in the outdoors, that it was effective even with shorts and not full clothing. All right, so that's the question on where ticks crawl and where they bite. The next question we have is, what is the success rate of a single dose of doxycycline after discovering an embedded tick and preventing Lyme disease or other diseases? And the second part is, any good reason not to give a single preventive dose of doxycycline immediately after discovering an embedded tick while awaiting your testing results?
So first, I have a disclaimer to the question. I am not a practicing physician. I am not able to treat or give out doxycycline prescriptions to anyone. I am a PhD, and thus but in my view, because I worked with animals as well as with humans, and I actually was an infectious disease doctor at Mass General Hospital, working alongside the godfather of vaccine development against Lyme, Alan Steer. He's my colleague. So I know a lot of people that are MDs that treat using doxycycline, and a lot of my friends are Lyme disease expert physicians. So in my response to that, if you have an embedded black-legged tick, and you're within that 24 to 72 hour window, preventative doxycycline is 100% effective to prevent Lyme disease and the other diseases. If it's a dog tick, it does nothing in New Hampshire, and because dog ticks tend to not be disease. So identifying your tick is very important. And if you treat with doxycycline preventatively for the tick that isn't carrying a disease or a dog tick, it can have an effect on antimicrobial disease resistance as well as your gut microbiota, because doxycycline kills all bacteria in your system and could harm your beneficial bacteria as well.
Okay.
Yeah.
All right. Well, I know that CDC did come out with some updated guidance on treatment of tick-borne disease. I believe it was 2-3 years ago. So that is available out there. If you search the web, and you might come up with an answer to that question for anybody who's listening in right now. We have another question, more specific to your expertise. Have you found any Lone Star ticks in New Hampshire?
According to our study from 2018 to 2021, we found a total of 7 Lone Star ticks in New Hampshire.
7. That's good to know and bad to know. What's the primary pathogen and health outcome from Lone Star ticks?
I don't.
Is that alpha gal, I believe?
Yeah, alpha gal, but they can also transmit other various diseases. Yeah. Okay.
So not many yet, but we're concerned, and people need to definitely pick their ticks off and report them.
BeBop Labs didn't do a very good method of tracking if people traveled out of state into New Hampshire and then found the tick when they were in New Hampshire. So again, the one dog tick that tested positive for Francisella tularemia, all the Lone Star ticks could have been a result of an individual traveling and finding a tick when they were in New Hampshire. Again, this is passive surveillance, so you have to factor that in.
Yep, and you're correct that every weekend, I don't know the exact number, but it's tens of thousands of people come from Massachusetts and other states, both to hike in the White Mountains and be around our lakes. So those people leave the state, go to other states, and that's where their data gets reported for the most part. And I'm not sure about what the interagency or interstate agreements are on trading that data. I think we have some with Massachusetts, but if anybody else knows about that, put it into the chat. All right, the next question is, are there any health disparities to be aware of with Lyme disease?
Yeah, I mean, we're in America, right? So there's inequality spread all over everything. Of course, there's health disparities for Lyme disease, and inequalities out there for low income and race. So like there's that Lyme disease rash, that the bullseye rash that people say is diagnosis. It's actually 100% diagnosed if you get a bullseye rash, but depending on the color of your skin as well as your immune system, that rash only appears maybe 20% of the time. So yeah, if you get it, it's 100% sure that you have Lyme. But if you have a different color of skin, you're probably not going to see it. So that's a big one too.
Good. So yeah, people with darker skin are going to have to really be tuned into their exposures because they're going to have one less diagnostic sign that they can key in on. I think the other health inequity I've thought about, especially in the North Country and around, is that outdoor workers, who are usually lower income workers, depending on what they are, certainly had a much higher risk for exposure to ticks and tick-borne disease. I haven't seen the occupational data on that yet, but those are people who were trying to do more outreach to in tick-free New Hampshire and trying to build up more instructors to go out and do training for outdoor workers. We're looking at the state of New Hampshire, specifically Department of Transportation and Environment, to make sure those people who might be at higher risk and have more difficulty getting to treatment know about the risks and know about the prevention. Okay, next question. What are some of the long-term effects of chronic Lyme disease, and what is the current treatments available, even if they're holistic? So take that first part. What are the long-term effects of chronic Lyme disease?
There's a number of symptoms for chronic Lyme. So Lyme disease likes to hide from your immune response in places of your body, but it also has a trick called antigenic variation where it modifies the way that it looks to your immune system. So the places that Lyme likes to hide also reflect your symptoms. So if it hides in your bones, you get Lyme-like arthritis. If it hides in your muscles, you get muscle fatigue and weakness. If it hides in your brain, you get brain fog. And people have done like necropsies on humans that have been chronically infected with Lyme, and the spirochetes will come bursting out when they like do necropsies. It's crazy how much you can hold and still live in your system and not get treated and somewhat function as a human. So the current treatment, so it's very tricky to treat chronic Lyme disease. There's several different methods, and it very much reflects personalized medicine. So the way that Lyme disease treatment is going is personalized to the individual, because every individual's immune response as well as status in their disease is different. So you take an individual approach, and the different approaches can include long-term use of antibiotics, herbal remedies that attack the biofilm, like curcumin, stevia, oil of oregano, all sorts of things have been used to attack the biofilm, and then in conjunction add an antibiotic. Sometimes it's other antibiotics that need to be used. Sometimes you have to have a blood perfusion to like attack any bacteria that are circulating in your blood to help decrease some of the symptoms too. So you really do have to have kind of a Lyme literate doctor to help with that. And it is treated, like it has been effectively treated. So it is not something, like it is something you can treat if you get the right personalized medicine.
Yeah, I think that one of the key things is if you've been to your primary care physician, you believe you're exposed to ticks and you might have a tick-borne disease, but you're, have these late symptoms, getting in touch with an infectious disease specialist is going to be key. They'll be able to guide you to the correct treatment, the correct assessment and treatment before you begin, trying any herbal or other medicines. That's the most important thing. All right, one of the questions, no, I think this is a thanks. That was thanks for the presentation. It's been fantastic. So we're at 7 o'clock and we've completed all the questions in our chat. If there's anything else, please put it in there, but I think we're ready for a wrap up. So I'll turn it back to Emily to do that.
Yeah, awesome. Thank you so much, Matt, for moderating and adding your very helpful input. And thank you so much, Dr. Morse, for this excellent presentation. I agree with everyone else when I saying that this was really informative and definitely really pertinent information for us all to hear. So I appreciate your time. I appreciate everyone else's time being with us tonight. I will follow up with some materials on this information about our next webinars, and to join as a member of New Hampshire Healthcare Workers for Climate Action. So thank you all again, and I look forward to seeing you sometime soon.
Great. Good night.
Thank you all.